Good morning or good afternoon, everyone. Welcome to Genome Webinars. I'm Julia Caro, Managing Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Multi-omics in Clinical Cancer Diagnostics, the Importance of RNA-seq. This webinar is sponsored by Lexogen. Our speakers today are Dr. Andrew Beggs, Professor of Cancer Genetics and Surgery at the Institute of Cancer Genetics and Genomic Sciences at the University of Birmingham in the UK, and Dr. Lucas Paul, Senior Manager of Scientific Affairs at Lexogen. You may type in a question at any time during the webinar, and you can do this through the Q&A panel, which appears on the right side of the webinar presentation. And we will ask our speakers your questions after the presentations have concluded. Also, if you look at the bottom tray of your window, you will find a series of widgets that can enhance your webinar experience. So with that, let me turn it over to Dr. Beggs. Please go ahead.
Hello and thank you. I'll just go to the beginning of my slides. Hold on. So I'm going to talk about the importance of clinical cancer diagnostics and the importance of RNA-seq, and thank you for that introduction. This talk is going to be focused mainly around colorectal cancer. I'm going to base it on a project that we carried out with Illumina UK and also later on with Lexogen, where we molecularly profiled a large set of colorectal cancers using whole genome sequencing and RNA sequencing, focusing mainly on the clinical side of it because that's the area of unmet need. Now, as you may know, colorectal cancer is a disease of unmet need. 40% of patients present with metastatic disease with few treatment options. And there have been quite a few advances in other cancers recently. For instance, in lung cancer with the lung matrix trial that I'll cover recently, it's been identified that comprehensive molecular stratification will cause substantial benefits to patients. But colorectal especially has lagged quite way behind because of, although we have a lot of data, we don't necessarily have the understanding of how these pathways all link together. Some advances have been made, so it's recently been shown in a niche study that PD-1 and PD-L1 inhibition is likely to cause a substantial regression of the tumour when given it in the neoadjuvant setting. And the Foxtrot study, which was presented at ASCO last year, showed that if you give neoadjuvant 5-FU and oxaliplatin for colon cancer, that improves prognosis, causes tumour to shrink, and makes disease more resectable. You've probably all seen this slide. The Chalabi et al. Paper in Nature Medicine of this year is quite earth-shattering in colon cancer world, showed that if you give neoadjuvant immunotherapy, you get profound tumour regression. And in the red bars, you can see that in patients with defective mismatch repair, unsurprisingly, if treatment with a combination of ipilimumab and nivolumab caused profound tumour regression of up to 100% in the majority of patients. And in fact, in my own practice recently, because I practice clinically as well, we've had a patient we've put through this regimen and showed complete tumour regression. But also, very interestingly, if you look at the green bars in the graph, there's also been some tumour regression as well, too, in patients who have proficient mismatch repair. And this presents quite a diagnostic conundrum. Obviously, there are some patients that benefit, but how do we identify these patients? More recently as well, ASCO the last year, Matt Seymour and the Foxtrot group presented data that's showing that giving neoadjuvant chemotherapy, a very simple one, oxaliplatin and 5-FU, increased response rates, decreased recurrence, and made tumours easier to resect. However, quite interesting, in a subgroup analysis they presented, there was no response to this chemotherapy, the defective mismatch repair tumour set, suggesting that these type of patients have a different molecular prognosis, and that a very simple marker, just mismatch repair immunohistochemistry, can stratify patients into ones that can have chemotherapy and ones that can't. And so it's quite important to identify these patients. And finally, I talked about the lung matrix study. Gary Middleton and his team here and Cancer Research Clinical Trials Unit looked at lung cancer. They recently published in Nature and showed that using rational agent selection, using a limited, I think it was 26 gene panel of next generation sequencing biomarkers, led to measurable patient benefit. But this was in the setting of metastatic lung cancer. And although patients definitely had benefit, there were small numbers in each group, as befits this type of study, but there were definite improvements seen. So there is a background to colorectal cancer. You may say, why have we done this study as the TCGA exists? The TCGA does exist in colon cancer, and it encompasses more than 600 cancers. However, a lot of the TCGA, with a few exceptions, were based on exome sequencing data sets. A lot of the mutations and insights that have been gained from this have come out of whole genome exome sequencing data. And although they've been linked with RNA-seq, the RNA-seq that was performed on the TCGA set and colon was starting quite a while ago. Technology has moved on since there. Also, there is whole genome sequencing data that exists for colorectal cancer and TCGA, but it's less than 50 tumours, and it's not well linked back to the clinical data set. Now, in the TCGA data set, they found differing molecular subtypes of colorectal cancer, as you have seen previously in other tumour types. They found a hypermutant type of poorly identified by Ian Tomlinson and others, the defective mismatch error part, and generally characterised patient samples into a hypermutated versus non-hypermutated group. And this is useful from the point of view of disease stratification. It didn't really elucidate much from the point of view of treatment options for patients. For those of you who are familiar with the UK landscape or based in the UK, you'll understand that the 100,000 Genomes project was a UK government commission project from 2015 to 2019. And the idea was to try and normalise, not so much as to do a large biological piece on whole genome sequencing in cancer, although that was the achievement, was also to normalise the use of whole genome sequencing as standard of care. But it was realised right at the beginning, actually, when we started doing this project, that patients who'd had whole genome sequencing performed as standard of care were missing an additional dimension, and that was RNA sequencing. So we were already commissioned to carry out an immediate pilot in association with NHS England based on a sample of convenience we had of frozen colorectal cancers, and to generate a set of whole genome sequenced colorectal cancer that could be linked back to other data types, such as RNA-seq and others. As I said, the pre-pilot data was a sample of convenience, and we took 54 sequential patients. We took fresh frozen tumour normal pairs with their colorectal cancer, and they underwent whole genome sequencing using the Genomics England pipeline that I'll cover in a bit of time. At the time, unfortunately, because when we started the project, we'd have thought that we would do fresh frozen whole genome sequencing. Nobody thought to preserve material for RNA-seq, and so actually, because the tumour biosamples were quite small, by the time we'd completed the project and extracted enough DNA, there was at that time, no fresh frozen material left from RNA-seq. So what was very fortunate for us is within the timescale of this project, RNA-seq using techniques such as Lexogen's QuantSeq off FFPE samples a possibility during this project. And when the tumour samples were collected at the time of project, we took what was called a molecular block, as is standard now with the 100,000 Genomes project. So in other words, the region that was sampled as part of the sample for the fresh frozen was also sampled into a formalin-fixed paraffin-embedded block that was available for use in the study. And we took these blocks and used them to sequence the RNA, and I'll describe some of the things that you can do with the RNA later on in this talk. So what was the methodology? I'm not sure how to reproduce for you all, but the advantage of the variant pipeline that was used for 100,000 Genomes project, and it has advanced a little bit since this, it's constantly evolving pipeline, is it was quite standardized. It was Illumina-centric in that a lot of the techniques and methodologies that were used for the whole genome sequencing calling were developed by Illumina internally, based around what they saw as the priorities for the 100,000 Genomes project. So both normal and tumour reads were aligned using their internal aligner ISAC. And for this project, we used version 3, however, it has moved on to version 4 since then. And then germline SNVs and indels were called using their caller Starling, which is based also on their caller Stroker, which was used to call the somatic SNVs and indels. And obviously, these callers have been used in head-to-head challenges before, and there are other callers that are better in certain scenarios and worse. But we wanted to use a pipeline-based approach that was going to be used in the real world. And so this variant calling methodology has been used now in over 30,000 tumour samples. And in the process, we found the advantages and the disadvantages. I'm not going to cover those today, but there are certainly strengths to using this approach. Structural variants were also caused using MANTA, which is the Illumina's structural variant caller, and CMV calling with Canvas, their copy number caller. They provided their own annotation pipeline, Nirvana, which is very good actually, and can be used to annotate the clinical variants. And outward, you'd get a summary as a HTML file, a PDF reports, and the VCFs that you could use downstream for whatever analysis you wanted to do. So this is integrated, it was pipelined, the data would stream off the HiSeqs onto the Genomics England computing environment, and then it would go through this pipeline in order to produce a set of clinically annotated validating variant calls. At the time of the study, the pipeline was research use only, but I believe now that NHS England and Genomics England have now successfully secured clinical ISO accreditation for the sequencing pipeline based on this methodology. So it's robust and reproducible, and it can be said to be clinical grade. As you can see here, we got good coverage from sequencing. Every sample was sequenced to almost 40x. As you can see, it's not completely over 40x, but it's definitely over 30x, and this was off germline samples. And so this provided high coverage in order to subtract the variants that we knew were going to be in the germline. And for the tumor, the standard was set at 60x for whole genome sequencing of the colorectal tumors. However, with fresh frozen material, Illumina managed to achieve mostly over this. Some of the tumors approached 90x, and we would always aim to go over at least 60x, if not greater. And so this provided a bit more flexibility to detect some of the lower penetrance variants that we would have seen in these patient samples, because obviously colorectal cancer is quite heterogeneous, and it's important to try and get a reasonable trade-off between read depth and sequencing costs in order to try and identify whether the variant's true or not. The downstream analysis meant you could do some pretty interesting things. For instance, this is a graph of the 1,000 genomes panel for ancestry mixture. Now, the sample set for which was taken on for the UK is quite diverse. We have a diverse population locally. As you can see, we managed to identify quite a set of different patients within there, some of East Asian ancestry, some of South Asian ancestry, but mostly of as it's put here, European ancestry, which sort of fits with our local population demographic. Now, it may not seem relevant, but actually, from the point of view of sample quality management, an ancestry inference, just in very general terms, can give you a bit of reassurance that you've not mixed samples up at a very high level. Obviously, we did this from the point of view of just understanding if there was any admixture in our cohort. And it just shows the power of whole genome sequencing data that you can get a lot of interesting other information out of it you wouldn't expect normally. Now, unsurprisingly, what we found in our whole genome sequencing cohort was that there was a clear binarization between what was termed in the TCGA, and we found as well, between hypermutated and non-hypermutated samples. And this are SNVs and indels, with the indels shown in red and the SNVs shown in blue. As you can see here, the non-hypermutated group had what we expected. There were some with very low numbers of SNVs and indels, and some were very high. As you can see here, the average was probably about 15,000 SNV and indels around the non-hypermutated group. And in the hypermutated group, there were a series of ones which was what we would call moderately hypermutated, and this patient here, who was ultra hypermutated, and I'll talk more about that patient later. But already you can get a useful clinical phenotype out of a whole genome sequencing data. You can identify the fact that there are patients there who need who may be suitable for immunotherapy. Now, one of the other useful things, and this is something that's slightly underdone, is the use of signatures in clinical data sets. They're quite important, I and we believe, and they can lead to quite interesting insights. Now, these are the mutational signatures. These are the SBS3 Sanger signatures by Alexandrov et al. That we've used through the mutational signatures package. Now, this isn't supported as a part of the primary analysis by Genomics England workflow, although it is coming online now, certainly not using clinical scenarios, but it is quite useful to get insights into the data you're looking at. Now, unsurprisingly, the most predominant signatures that we found in colorectal cancer were aging, tobacco, and aging, 155 and 40. And this is because colorectal cancer predominantly, unlike some other cancers, say melanoma, where there's a UV signature, is a disease mainly of the late 60s to 70s. That's when most people develop colorectal cancer. So it's useful that the aging signature was found in these tumors. It sort of fitted what we understood about the biology of them. Unsurprisingly, we also found other signatures, such as the defective mismatch repair signature 44, which was reassuring because that linked up nicely with what we'd seen in the patients with defective mismatch repair. An ApoBec signature was interesting. It has been described in colorectal cancer, but not like in breast cancer where it's more frequent. Pre-treatment with 5-fluorouracil, so there were 2 patients within this trial who'd been through the Foxtrot study you'd seen, so they had neoadjuvant 555FU, so that was quite useful as a sense check that we could identify them. And also the combined POLD1 and defective mismatch repair signature, which none of you will be surprised to learn was the one with the ultra hypermutator there, because as we know, these POLD1 patients and defective mismatch repair can have co-mutations that cause a very hyper supermutator phenotype. Now, interestingly, in a cohort like this, we didn't see any germline mutations. Now, and what I mean by that is there are a series of germline mutations that predispose people to colorectal cancer. in the mismatch repair genes such as MLH1, MSH2, MSH6 that act to cause defective mismatch repair, and also the genes identified by the Tomlinson group and others, POLE and POLD1, which cause a hypermutated phenotype. And in a cohort like this with just over 50 patients, we would have expected to see 1 or 2 patients who have a Lynch syndrome predisposition. I can tell you that from the 100,000 genomes work that we've done locally, that approximately 4 to 5% of people we sequenced had Lynch syndrome predisposition. And we think this is probably because we were quite good at identifying the patients who went into this. People, as standard of care, patients who come through our institution will have mismatch repair and immunohistochemistry, and we'll know whether they are likely to have Lynch syndrome before we sequence them, because they'll also have BRAF mutation status and MLH1 promoter hypermethylation status. So from this, we deliberately made an attempt to exclude any patients with Lynch syndrome that might go in. But at the time we did this study, the proofreading polyposis syndrome genes, such as POLE and POLD1, were not massively widespreadly known, so we didn't filter any people. But once again, we didn't find anybody with a germline POLE or POLD1 mutation. But we did find people with somatic mutations in all of these genes, which was reassuring. And luckily for us, given that somatic POLE mutations are quite rare, and POLD1, we found somatic mutations in the classical hotspots in both POLE and POLD1. And this was useful for us because it allowed us to understand whether our variant calling pipelines could stand up to calling hypermutated samples. As you can see here, the mutational spectra is kind of what you expect in these kind of patients. And the mutational biases. There was a CTT bias, which was slightly interesting. We assume that's from the aging part of the mutational spectrum, because obviously normally you would see a CTT bias in FFPE samples, but obviously non-FFPE samples were done here. So we think that's related to the aging phenomenon that we saw. In terms of colorectal cancer driver mutations, there were unsurprisingly quite a few APC mutations. As we know, this is the predominant driver in colorectal cancer, but interestingly, we only saw 66% of patients had APC driver mutations. Now, this sort of fits in with what we understand about colorectal cancer biology. It's usually a defect in Wnt signaling, so things like R-spondin mutations, RNF43 mutations, which you can see here were present in the patients who didn't have an APC mutation. But there were some people who, using this pipeline, didn't have a detectable mutation in the DNA level in one of the Wnt driver genes. And interestingly, in these people, there is usually an R-spondin fusion. So we had a slightly higher preponderance of R-spondin fusions than we were expecting. Otherwise, we also saw driver mutations that were classically associated with colorectal cancer, such as KRAS, POLE, POLD1, and others within this cohort. And we also found a high frequency of TP53 mutation, which was reassuring. Now, what we did then is, one of the ideas around this project, because it was a sample of convenience, is could we understand the utility of these driver callers? There are software packages available that can look at the mutations that are pulled out of this kind of sequencing analysis and use them to draw them together to say, well, this is likely to be a driver. And we used multiple callers during this project. Some of them are better than others, so MuseCV2 done by the Broad, Integen by the group in Barcelona, FunSeq2, which we also use from the point of view of non-coding drivers, and DND-SCV, which is written by the Sanger. And we combine the output from all of them, see if we could identify novel drivers and confirm existing ones. As you can see here from this Venn diagram, I have got to give credit to Venni. It's a very good online tool to make these kind of Venn diagrams where you can paste gene lists and get overlaps. We found that each profiler, unsurprisingly, which shows the weakness of some of these algorithms, produced different mutational drivers compared to the others. And actually, only the overlaps of the 4, the only one that showed an overlap was this one here, which unsurprisingly was APC. And that was known as a driver of colorectal cancer. MuseCV tends to be quite noisy. And so when we removed the MuseCV calls, you can see here, we found the classical drivers, so KRAS, TP53, FBXW7, an increasingly important tumor suppressor gene, and one that my lab has an important interest in as well, and PIK3CA, which was known about. We also found some novel genes. Now, they're known to be associated with cancers, but they're not really classically associated with colorectal cancer. And these need further exploration, such as BCL9 involved with apoptosis. MSH3 has not really been described as a classical Lynch syndrome predisposition gene, or even a driver gene within colorectal cancer, so it'd be interesting to understand that. There is some evidence that it causes a hypermutated phenotype. Delta-catenin as well is another one. The beta-catenin is obviously part of the Wnt signaling pathway, but delta-catenin, which signals off it, is another one that's been shown up. So these need to be explored further. But the fact that they've come out of three different driver callers is reassuring that there may be something going on here. So we also performed a copy number analysis, and this was based off binning of the whole genome sequencing data with short reads. Obviously, this is advantageous for calling copy number data. And what you can see here is a heat map plot of essentially the frequency of the copy number gain loss. We're 100 up there at the top, 100% and minus 20. That's the losses there. As you can see, some of them are virtually ubiquitous to have copy number loss, not completely, but some are virtually ubiquitous, which sort of fits with what we know around colorectal cancer, in that some colorectal cancers have losses seen in these regions, which are thought to be important, but nobody's absolutely sure yet. Unsurprisingly, we saw that over 50% had a loss of X, obviously because about 50% of the patients in this study were men, and so that's why we expect to see the loss there. And when we looked at across the whole cohort, we found an easier way to do it, assuming that the copy number changes we would see within the cohort were pathogenic in the protein coding regions rather than in the so-called gene desert regions. We filtered by exome positions. And when we filtered in exome positions, when we sort of set a threshold of over 80% of samples having a copy number change, and we limited it to ones where there were changes within a coding region, we found that one of the 4-KET binding domain genes, FOXY2, had a recurrent copy number gain, and that REX1BD, which is a part of a DNA repair excision pathway, also had a recurrent copy number change there. So once again, we haven't explored these yet, and this type of data set, one of the issues with it, provides a rich variety of potential findings that could be the subject of a research project, any one of them at all, really. So it just shows already with copy number analysis what you can find. There are also other phenomena I'll just briefly touch on that we've seen in cancer, and for instance, the phenomenon of double minutes within colorectal cancer. This is a relatively genomically, chromosomally unstable colorectal cancer here, as you can see in this B allele frequency plot in the blue, and above it in the red is the log R ratio, suggesting things like gains, and you can see here there are gains in two of the chromosomes here. And the phenomenon of double minutes you can see here, localized between chromosome 13 and chromosome 20. And there are a number of high copy number double minute genes that are likely to be associated with cancer predisposition. So Aurora kinase A, the transcription factor SRC, BRCA2, which we all know, and the FGFs, the fibroblast growth factors. Now, it's difficult to know whether these fibroblast growth factors are actually what we're seeing within the tumor itself or with any associated stromas. And so obviously it's important to, and one of the things that this study doesn't take account of is tumor heterogeneity, as it's difficult to tease out in the samples that are taken for this type of projects. But it just shows that with whole genome sequencing data, you can pull out a lot of interesting findings already within this type of methodology. We've also detected catalysts, which is detected previously in breast cancer. These are mutations of localized somatic hypermutation. And we've shown within this sample here that there were 39 breakpoints within this 7.3 megabase region where there was quite a lot of somatic hypermutation, suggesting that there was something going on here. And this can be detected computationally as well, and is another interesting finding showing that this phenomenon occurs not just in breast cancer, it's also been seen in lung cancer and now also in colorectal cancer. And it's only really whole genome sequencing that can pull this out. You can try and identify with exome sequencing, but it's a lot more complicated than would normally be the case. So another fascinating insight using whole genome sequencing. Now, another thing that is interesting about whole genome sequencing is the ability potentially to detect gene fusions. Now, normally RNA sequencing would be used to do this. And one of the problems we had with this study, as we've discussed already, is that a lot of the source material had been used up and we only had formalin-fixed material afterwards. So we went to use, and the bioinformaticians working on this project, used an approach to split read approach, unsurprisingly, to detect potential gene fusions using whole genome sequencing. And there was a number of gene fusions that were identified using this approach. And as you can see here, BRAF is a critical gene in colorectal cancer. It's mutated in about 10% of colorectal cancer. It's usually a hyperactivating mutation. And this is a fusion between BRAF and DLG1, which are different chromosomes, and it makes a fusion transcript. Now, the question here was this real or not? So we did Sanger sequencing across the breakpoint, across the fusion boundary, in order to see whether it was genuine. Yes, it did seem the whole genome sequencing, and this was a 125 paired end sequencing strategy, could detect fusions reliably in this data set, which is great, really. We weren't expecting it. It's not 100% accurate, but WGS fusion detection is a possibility with this kind of data set and is another thing that enriches it. I would always advise, though, from what we've seen, that you should validate this fusion with a downstream orthogonal technique, as it's not 100% accurate. If you're going to make clinical treatment decisions based on this, then you should really use an orthogonal technique for the time being, especially with whole genome sequencing data. Now, one of the other things that's I think was really interesting and really cool, now this is also fraught with difficulty, but this is more of a proof of concept analysis. We performed a metagenomic analysis. Now, as you know, with whole genome sequencing data, about 5 to 10% of whole genome sequencing libraries consist of all the other stuff that goes in with the tumor. So the bacterial reads, the viral reads, the fungal reads that will go in with it, that are part of the tumor, but also contaminating the kit. And we know what kit we use. We've read the kitome papers, so we sequenced the kitome essentially and used it to make, and also used the kitome papers to take a list of things that we know are contaminants and take them away. But essentially, the way we did a shotgun metagenomic analysis of this is we pulled the unmapped reads from the BAM files using SAM tools. We converted them to FAST using Picard, and then we did a very simple analysis. It's not what I would describe as a proper metagenomic analysis, and I know a lot of metagenomicists will shudder when they see this, but it's just a proof of concept that you can detect biologically relevant metagenomes within these cancer samples. And we use Kraken 2 because it's very quick, and we use Pavian to do the reporting, as I'm going to show you here. So we saw Fusobacterium in the sample, which is been well known now for years to be associated with colorectal cancer. Correlation doesn't mean it's a valid methodology, and so obviously this needs a lot more development. But we also detected many reads in things like Klebsiella pneumoniae, E. coli that we know live in the gut, and so it was reassuring that we were detecting genome reads here after the kitome had been sequenced. So that's great with whole genome sequencing. You can get all these methodologies. You can get the SNPs, the indels, the germline data, copy number, structural variants, metagenome, and also other interesting biological insights to the tumor. But it doesn't quite give you the depth of information you want. And we now know from many research studies that the extra dimension that we need is RNA-seq. As I explained, we struggled a little bit with the fact that a lot of material had been used up. So we went towards another technology, and very presciently at the time, Lexogen came to market with their 3' RNA-seq technology, QuantSeq. And we used the FFP genomic block that I described before, so it had come from the same part of the tumor that the fresh frozen sample had come from. We used 50 nanograms of FFP-derived RNA, although we can and we have internally here in Birmingham gone down to much lower than this. In fact, we've successfully prepped RNA-seq libraries with QuantSeq down to 500 picograms of FFP RNA. But that presents with its own problems, and we use a NextSeq and we pulled it onto a flow cell. And so the advantages to us, well, the library preparation was quick, it worked off the material we had, and it was cost effective for us. Others and ourselves have done head-to-head validations on other tumor types for this and shown good correlation between the fresh frozen and between the FFP samples that have been done. And so we were confident going into this that what we did was likely to give the results that reflected what we were going to see in the fresh frozen tumors. Interestingly also, one of the criticisms of FFPE sequencing has been, well, the material you get back is terrible, it won't sequence properly, and certainly when we used to do the old style of FFPE sequencing where you would do a total RNA-seq prep off a large volume of FFP-derived RNA, that was the case. You'd have low read alignments, you'd have a lot of unaligned reads, you'd have a lot of chimeric reads, and it was difficult to pull any sense out of it, and you'd end up spending a lot of money not to get very much answer. Now, the good thing about the QuantSeq, this is a graph of the alignment quality. On the y-axis here, this is the percentage of reads that are aligned, and on the x-axis is just different samples. And the green represents perfect alignment, the yellow represents more than one alignment, and the red represents no alignment. So as you can see here, actually, the majority of the reads mapped back to the what we would expect to be in the host genome. So that was fantastic from our point of view. We were really pleased with how this worked, in that the quality of alignments we saw from FFP RNA, and don't forget this is at the gene counting level, the transcript counting level, rather than the full length transcript, was good enough and really pleasing for us to do what we needed to do. Now, one of the issues you do have with QuantSeq, and it can cause problems, but there are ways around it, is what I've put read depth here. It's essentially number of reads. And although we aim to pull for over at least 1 million or 2 million reads, I mean, how many you do really depends on what you want to find. As you can see here, there's one sample here where we had very few reads. And this, in the end, turned out to be a library hybridization problem. So when we repeated it, as I haven't shown that here, it did improve the sample such that we could sequence it successfully. But we do get dropouts occasionally. Usually, the best methodology we found to get through this is re-extraction and re-sequencing on a separate library, and it usually comes back. But there are some samples that aren't even rescuable with Lexogen RNA-seq, and so they remain out of reach, unfortunately. But generally, we're pretty pleased with how it works. Now, another advantage of the Lexogen RNA-seq is it comes with validated and containerized workflows to do your downstream analysis. So various different ones. Lexogen use their own Bluebee. You can use Partec Flow. You can design your own, but there are multiple ones. And certainly when you're doing clinical grade RNA-seq, is what we were intending to do, because our entire pipeline is now validated and accredited at ISO level, is that we want to try and standardize everything that we do. And so now we use a standardized RNA-seq alignment pipeline in order to call gene expression in these samples. But the downstream analysis is what's interesting. In the past, you know, the TCGA have pulled out different transcriptional subtypes. Others have pulled out other transcriptional subtypes I'm going to discuss in a second. But we performed, just an interest to see how the data clustered on its own, we performed hierarchical clustering of the most 100 variable probes. And we detected 5 clusters within this RNA-seq dataset. I haven't gone into the data in depth here, but there was no distinction in terms of site, in terms of mutational drivers. They clustered into interesting groups that didn't seem really to have anything to do with the classical classifications of colorectal cancer. So, for instance, we looked at, so I'll just go back, we looked at, we found that one of the clusters was TGF-beta dependent. We found that one of the clusters was immune. Unfortunately, there was only one sample in clusters 4 and clusters 5, and so it's difficult to work out what the cluster was in there, but it does show that you can detect clusters within it. Now, one of the other things you can do as a surrogate marker for immune infiltration is use CyberSort. Now, CyberSort, for those of you who don't know, is an algorithm that effectively deconvolves RNA-seq data, given if there's sufficient of it, in order to identify the immune cell infiltration profile within cancers. Now, this is enormously powerful. One of the problems with histopathology, I hope there's no histopathologist listening, so I may chuckle as this, is generally histopathologists can't always agree with each other about how many tumor-infiltrating lymphocytes there are within the sample. They'll say it's rich, but it's difficult to quantify exactly, and it's also difficult to differentiate apart from the tumor-infiltrating lymphocytes. So we use CyberSort to deconvolve our RNA-seq, and I haven't shown it in the graph here just because of time constraints, but we found that the tumors that had hypermutation had a high score for TD4 and CD8 infiltration, unsurprisingly, but we also showed that there are ones that were relatively dead in terms of immune infiltration, which is quite useful, as this might end up being a surrogate marker for something we can use for immunotherapies in the future. But we also, this is just a summary presentation of the different patients within the cohort, and it showed that obviously, unsurprisingly, we found a lot of memory resting cells, but interestingly, we found a lot of M2 macrophages. And macrophages have been highlighted as being an important immune infiltrating cell in colorectal cancer. It's unknown as to what the role they play in this, but the fact they're there and in relatively high numbers is quite interesting. And what we did as a sort of sideline is we looked at some of the H&E slides of these tumors and showed that the CyberSort had a reasonably good correlation with the H&E as others have shown. There's also some activated dendritic cells in there, which is particularly interesting for us. Dendritic cells are known to be an antigen-presenting cell. What does it mean? Difficult to know. And well, as I said, once again, with the RNA-seq you do, it can help you enhance and expand what you're doing. Now, as I said earlier, with the pathways analysis we did, we found a variety of different pathways within each cluster. There was, within cluster 1, there was inflammation and DNA repair, which sort of fits with the general hypothesis that colorectal cancer is a disease of aging. We had Th1 and Th2 centric group in number 2, IL-17 driven, which is interesting because that might be one that could be targeted by immunotherapy. And that was a relatively small group. Within group 3, which is a slightly larger group, that was very much driven by Wnt and BMP signaling, and that would fit in with the Wnt-addicted tumour hypothesis. So some colorectal cancers are thought to be Wnt-addicted, and certainly we wonder whether this might be the ones there. And there have been a paper recently that I was on showing that these patients may respond to TGF ligand manipulation. As I explained earlier, there was no real correlation between any pathways in 4 and 5, this because we only had one sample in 4 and 5. Now, this may be a problem with classification, but as you can see here, though, hopefully, between cluster 4 and 5, they were transcriptionally substantially different between the two sets. And so we would probably need to understand a larger cohort in order to understand what's going on here. Now, one of the other things you can do with 3' RNA-seq is metagenomic analysis. I know I keep banging on about it. Now, we didn't think that metagenomic analysis would be possible on 3' RNA-seq data. The reads are relatively short. They are 3' biased. They will only work on polyadenylated for bacteria, fungi, and viruses. Obviously, not all of them are. Very few of them are. But it should be able to achieve metagenomic analysis. So we use the same type of pipeline. We took the unaligned reads. We used Samtors, Picard, and Kraken-2 to map them back. And we identified, once again, known pathogenic bacteria. So Fusobacterium, Bacteroides fragilis, bacteria, other Bacteroides species. And one of the criticisms that could be leveled is, well, how do you know this is just not contaminants? Well, we did sequence normal tumor within, normal mucosa as well as part of this, just because we were interested in the expression profile of normal. And we showed that what we saw in the normal colon was substantially different and clustered out from the tumor. What we saw in the normal is what we normally see in a normal gut, like lactobacillus, enterobacteria, stuff like that. So it seems to suggest that we could use 3' RNA-seq for metagenome analysis, and certainly that would be another significantly attractive advantage. It's something we could do in the future, but we need to do further exploratory work of this to understand its utility and also demonstrate its versatility and accuracy. Now, the other good thing about RNA-seq is you can detect alternate splicing events, and with 3' RNA-seq, it may be difficult, but actually with the read lengths, we demonstrated you could pull out, and we verified this we're downstream by resequencing alternate splicing events. And we found reasonably relevant alternate splicing events within the colorectal cancer cohort, so things like polycomb repressor genes, inhibitors of apoptosis, the respiratory chain genes, and methyl tetrahydrate folate reductase, which is particularly interesting because this is key in terms of glutathione and methylation, and maybe one of the drivers that is associated with one of the phenotypes of colorectal cancer. Showing that we could detect alternate splicing with 3' RNA-seq was quite interesting, quite attractive to us. Now, also, what's even probably the most important in terms of clinical utility of RNA-seq, and why is it important to add on, are things like the CMS and CRIS classification. Now, these are relatively recent classifiers that have arisen out of microarray datasets in colorectal cancer. The classical one is the CMS classifier, the consensus molecular subtype, by Justin Grinning et al. It was in Nature Medicine, and they performed a unsupervised hierarchical clustering analysis on colorectal cancer microarray samples to try and de novo generate molecular subtypes. Now, these are important because these have been associated with differential prognosis. For instance, if you have a CMS4 positive colorectal cancer, this is associated with a very poor prognosis because it's much more aggressive. It's put mesenchymal here, and it is primarily driven by what's called aggressive cancer fibroblasts. Whereas if you've got a CMS1 tumor, these are the ones that tend to be MSI high, they're hypermethylated, and they have immune infiltration. They do better if they treated well, but if they relapse, they do worse. So these CMS classifiers can give you an insight into the biology, but also the prognosis. And as you can see here, that CMS2 is what's known as the canonical pathway, so that's Wnt signaling activation, and copy number alterations are high in this. So can you predict these with 3' RNA-seq off these samples? The answer is yes, you can, because we'll show in a minute. But there is also one of the other things that's important is we've shown that you can also classify CRIS classification. Now, CRIS has also been shown to be associated with prognosis. As you know, CRIS was an evolution of CMS in that it was shown that CMS was substantially biased by the tumor microenvironment. So in other words, what you were seeing was a combination of the tumor microenvironment and the tumor itself. So for instance, if you had a very fibroblast-heavy tumor, it may classify it as CMS4, where in fact it may be a mixture, and it certainly fits into the tumor heterogeneity line. So the CRC intrinsic subtypes paper was published in NatComs in 2017, and this was based on purification of the tumor and then analysis this rather than the tumor microenvironment. And some of the CRIS classifiers have been linked to responsiveness. So some of them, for instance, CRIS-C will have a better response, will have a variable response to cetuximab, whereas some of them will have variable response to other agents. And so these are potentially important signatures that may be used in clinical practice in the future. And certainly from our point of view, it's quite useful to have a CMS and CRIS classifier going forwards. I think further work will need to be done to understand whether these are relevant in clinical practice. But for instance, a consortium that I'm involved in, the Stratification in Colorectal Cancer Consortium, ESCORT, has classified over 2,000 colorectal cancers from clinical trials for CRIS and CMS. And there are already associations coming out between prognosis and therapy and the CMS and CRIS classifiers. So these may well be increasingly important in the future. We showed that we could reliably and robustly classify CMS and CRIS classifier using 3' RNA-seq. And we showed what's been expected across the cohort, and then we saw a mixture of tumour samples. If anything, we saw a bias towards CMS4 a bit more in our cohort. It's difficult to know whether that's a true finding, whether it's biased by tumour heterogeneity, but we found the classifier algorithm is quite easy. For those of you interested, we use the CMS caller algorithm in our bioconductor. It's quite easy to export the gene counts from whatever piece of software you use and import them into this algorithm, and it rapidly and precisely generates a CMS classifier for you. And what we've done is we've integrated that back into our pipeline now, and every patient sample, in addition to having that, will have signatures derived. Now, it's likely that other people will have favorite signatures, and some signatures may be associated with prognosis. Ourselves, one of our PhD students at the time, who's now a lecturer in our laboratory, along with Gary Middleton and Ben Wilcox and others, made a classifier called the Kirk, which is a Th2-centric class 2 type signature for immune responsiveness. And we termed it the coordinate immune response cluster. And this was published in Oncoimmunology several years ago. And the reason we're interested in this signature is it's not associated with things like hypermutation and normal immunovisibility, but it's associated with immunovisibility that may respond to PD-1 blockade. And this is a classifier based on a 47-gene panel that was pulled out based on rational gene selection. And you can use it, once again, using that, you can adapt the CMS classifier algorithm to call a Kirk. And ourselves and others have shown there may be an association between the Kirk and immunovisibility out with things like hypermutation and PD-1 expression, and independent of tumor mutational burden. So these patients may be suitable for immunotherapy. Now, generally, this signature may not be useful taking forwards clinically in the future, but it's an illustration of the fact that you can very rapidly take this 3' RNA-seq data and you can use it to generate insights that may be useful for your clinical data set. And RNA-seq is enormously powerful to do this. You can rapidly adapt your own signature or clinical classifier of use and take it forwards as part of a whole patient approach with whole genome sequencing, or the copy number structural variants you've seen, and RNA-seq. And as we're moving more towards panel-based rather than our WGS at the moment, combining something like a large gene panel with instead of WGS with RNA-seq is both cost-effective and enormously powerful. So going forwards, I think, and we believe that RNA-seq should be added into standard of care as long as you've got a validated pipeline. So in conclusion, we think that our whole genome sequencing and RNA-seq is powerful. It allows you to gain some really interesting insights into these samples. And using a clinical pipeline, it allows you to access difficult samples. And I have to say, the Lexogen, and they haven't paid me to say this, the Lexogen pipeline is extremely useful. It's allowed us to access samples that are previously unaccessible. It allows you to understand a comprehensive examination of the entire biology of the tumour. And going forwards, it's not just used from the clinical point of view, but we've actually used it now from the preclinical point of view. So if you have a historical tumour set that you want to analyse in terms of thousands or hundreds of samples, you can do that. And we have used Lexogen methodology to do that. And it allows you to access these patients and these tumour types rapidly, robustly, and give you useful data that you can use. And I've used Lexogen QuantSeq data in order to buttress grant applications. And going forward for us, it will be the methodology of choice, for instance, in the recently commissioned sarcoma accelerator, where we're going to molecularly profile over 2,000 soft tissue sarcomas using this technology. So it's a fantastic technology coupled with something else and allows you really interesting insights. So thank you for listening. I'd just like to thank my lab here. As you can see, some of them at the motley crew at the top of them. I'd also like specifically to thank for the whole genome sequencing project, David Bentley, Mark Ross, and Meow here at Illumina. Meow did a lot of the bioinformatics work around the whole genome sequencing data, and she's worked really hard on this. I'd also like to thank some of our funders here, Cancer Research UK, the Wellcome Trust, MRC, EU, Sarcoma UK, and the Hospital Charity, without none of whom this work could be done. And that's it. Thank you very much.
Yeah, thank you very much, Andy, for this nice presentation. Just a quick reminder, if anybody has a question, please type it into the Q&A panel. So our next speaker is Dr. Lucas Paul. Go ahead.
All right. Thanks a lot, Julian. Thanks especially Andrew for this fantastic talk and insight into how RNA sequencing can drive forward cancer diagnostics. But to do so, this RNA sequencing method has to be sensitive. It has to be capable of handling also FFP-derived RNA samples or degraded material also from biofluids. And it has to be high throughput suitable and also highly affordable. And that brings me to what Lexogen provides. Lexogen is a complete RNA sequencing solution provider. So we provide kits and of course services from RNA sequence, from the RNA extraction, selection and depletion to NGS library preparation. And these NGS library preparations you can classify in terms of expression profiling. So Andrew talked about data derived from the QuantSeq 3' mRNA-seq library preparation kit. And I will introduce you shortly to a new kit that we have here, which is called QuantSeq-Pool. But we also provide for whole transcriptome analysis, small RNA analysis, and full-length cDNA analysis, of course. So QuantSeq itself is a tag profiling method, meaning that you get one read per RNA. And that is very powerful in terms that you get a very robust protocol. You just have to simply map back to the genome. You can count these mapped reads, and that is very straightforward also in terms of data evaluation. It's very user-friendly. You've got only very few protocol steps. You can use, as I mentioned before, all kind of samples down to 0.1 nanogram of total RNA input and also highly degraded samples. There's a high level of multiplexing possible also with the standard QuantSeq method, resulting in time and cost saving. And it's a highly established method. So there are already more than 600 publications citing it. And QuantSeq actually is part of a whole product line and a family. So we do have the forward version, the standard version for expression profiling, where you get the read towards the 3' end. There's the reverse version where you pinpoint the poly-A site for poly-A site analysis and quantification. We do have the QuantSeq Flex member of the family for targeted mRNA sequencing. And we have several modules like the unique molecular indexing module if you want to take care of PCR duplication. If you want to analyze whole blood samples, you might want to also block globin mRNA from entering the library prep, and you can do this with the globin block module. And of course, we take care of all your indexing needs with dual indexing and UDIs provided here. So some of the very positive aspects and advantages of QuantSeq are because it takes care of the sample input individually, meaning that you can have samples of varying quality. I'm sure that the FFP samples that Andrew's group had to analyze were not always yielding the same RNA amount and also were different in the RNA degradation status. And QuantSeq has a qPCR and quality control step included to take care of this. Now, there are setups where you have a screening approach to it, meaning that you've got a homogeneous RNA input like from cell cultures. And for this, we have developed QuantSeq-Pool. So the ultimate goal is, of course, always to make sequencing accessible, especially in the clinics. And in this kind of projects, you want to really drive down not only the direct kit costs and the sequencing costs, but also the associated costs. And this is now really possible with development of these new high throughput machines like NovaSeq, where you can get up to 10 billion reads from one flow cell. So translating this to QuantSeq, where you need about 3 million reads per sample, you can multiplex up to 3,000 samples now per run, and that can really drive down the cost considerably. We've taken care of this by providing a barcoding capacity of up to 36,864 samples with QuantSeq-Pool. So if you have got samples of varying quality or really low input amounts, so you can go down to less than a nanogram or a couple of nanograms for FFP samples, then the standard QuantSeq method is still the method of your choice. If you got a homogeneous input, then QuantSeq-Pool provides several advantages in addition, actually. The workflow is straightforward. You start with oligo-DT priming. That priming step already introduces not only the barcode, the I1 barcode as we call it, but also already a UMI tag. So UMIs are integrated part of the workflow already. Then after you have already sample barcoded at the cDNA synthesis step, you can already pool for purification and second strand synthesis. You then also do the PCR amplification, where you can add additional barcodes like the I5 and I7 barcodes. So by this, you can manually prepare 96 libraries in less than 5 hours, and you can very easily scale this up manually or also on an automated platform like the Kingfisher one to up to 36,000 samples. And you don't need to worry about preparing all those samples individually. You can pool at the second strand synthesis stage, and that results in a highly efficient workflow. Sensitivity is on par with QuantSeq standard, meaning that from 10 nanogram reference RNA, you can detect 7,500 genes with only 100,000 reads already. And the time saving is considerable. I mentioned you can prepare 96 samples in less than 5 hours because for homogeneous input, you can skip all those qPCR and quality controls. And that also means you can prepare 1,000 samples in 2 days. And that's also possible in a manual setup here. So the last slide here just illustrates one aspect of the money-saving advantage, and that's not the direct kit cost, but you save especially in terms of plasticware and quality control costs. Okay, with this I want to finish. There's much more to say. You can find all the detailed information on our website, lexogen.com, and also the contact details. And with that, I want to hand back over to Julia. Thank you.
Yeah, thanks very much, Lucas, for all the technical detail. As a quick reminder, again, if you have a question, please type it into the Q&A panel. And we would also like to ask our participants to take a moment after the webinar has ended to take our exit survey and provide us with your feedback. So let's see what we have in terms of questions. Yeah, a quick one for Andy. When approximately do you think the data you showed in your talk will be made public or published somewhere?
So the manuscript has been provisionally accepted subject to revisions in, I can only say a high impact factor journal, very high impact factor journal, because I don't want to bias the editorial process. The plan is that I will push the button to go live on the EGA, the European Genome Phenome Archive, once the data is once the manuscript is accepted. It should be we're making it all available publicly.
Okay, very nice. Yeah. Yeah, you talked a lot about the synergy between the RNA-seq and the whole genome sequencing data. Somebody wants to know whether there are examples where RNA-seq alone has generated insights that are useful for drug development without the use of whole genome sequencing, and why there is this synergy.
I would always say that you should do both. The reason being a lot of drug development has gone stalled off the fact that whole genome sequencing or any sequencing data has been used without RNA-seq data. So mutation may not actually be pathogenic, even though it appears to be. It may not cause a change in gene expression or pathway alteration. And the converse is true. There may be a pathway alteration with no obvious cause, and it may not be druggable. And also, if you're planning an experiment where you're going to do a time course with or without drug, I would always advise both, so you can look at selection pressures. Because in the absence of most modifiers, there has to be some kind of mutational change or evolutionary change for drug selection. And so it can be difficult if you don't have both data to look at. It can make things confusing. You regret it, and then you end up going back and doing it anyway. Right.
Okay. Yeah. And you talked a little bit about the dropouts for the RNA-seq. Can you just say a little more about that? Were there any dropouts? And can you comment on the quality of your FFPE samples in general?
So I would say our quality of the FFPE samples was terrible in the nicest possible way. The main reason being because at one point some of the samples had come from blocks that were not stored in neutral buffer formalin. So one of the things that's critical, as we've discovered, to a good genomic analysis is the use of neutral buffered formalin. And if you use old-fashioned formalin like we used to, it would destroy the blocks, or at least for mutation analysis. What we found with Lexogen is that that didn't seem to impact us as much as it had done with mutation analysis. So the quality going in was pretty terrible. The dropouts tended to be problems that when it was usually our fault. We'd not done the library prep completely properly. We'd missed out a sample, pipetting errors, things like that. So one of the things that's changed that for us is since we've moved over towards an automated, we have a Hamilton NGS style robot, and we've moved our Lexogen preps, if we're doing large scale ones anyway, over to that robot, and that eliminates those kind of small errors that can irritate you because you get one you get one sample that doesn't work in an entire run, and it's quite frustrating because then you think, when am I going to run that? So yeah, moving towards automation has helped from our point of view of improving things. But it's generally usually a problem you've had. We've not really, unlike other RNA-seq library preps, ever really had a full fail off this that's sample related. Usually we get some usable data.
Okay, thank you. And I know you said during your talk that you did not look at tumor heterogeneity. You looked at bulk samples, right? But somebody wants to know, yes, is that possible? Is that something you're considering to do single cell?
Yes, no, so we are funded to do that of colorectal and sarcoma, actually. So we have already done quite a bit of preliminary data that we are finishing at the moment. We're funded as part of what's called the Sarcoma Accelerator, which is an international project to carry out single cell sequencing data on colorectal and sarcoma. We're doing it on. One of the other things we've actually done is there's a recent paper published by a French group where we use 10x chromium to do that. Sorry to talk about another technology, but we use single cell, and when we are doing mutational signature with nanopore sequencing off the single cells. So that's going to give us a really interesting counterpoint to what we're seeing in the bulk sequencing data.
Okay, thanks. Here's the question that relates to the molecular subtypes of CRC. So the question is, were the samples you used following the distribution percentages for the 4 different known molecular subtypes? And did you think your findings were influenced by the possible dominance of one specific of those subtypes? And also, do you expect identifying novel subtypes if you focus on one molecular subtype? I hope that makes sense.
Yeah, it does. I read the question. So the answer is, for CMS, yes, we saw a few more CMS4s than we were expecting, which was, I suppose, not a surprise because they're bigger tumors, they're more aggressive, they're easier to sample. For the CRIS was almost exactly what had been published in the literature before. Did it bias what we found? Well, I mean, the CMS4 tumors are the mesenchymal subtype. They're the aggressive fibroblasts. So not really, no, from the mutational point of view. It might have biased the RNA-seq work. In terms of novel transcriptional types, as anybody who's done CMS and CRIS knows, there's always about 10 or 15% unknown or not classifiable. So there probably are more molecular subtypes out there that we just haven't identified yet. And I definitely think this is a work in progress.
Mm-hmm. Okay. Can you talk a little bit about your sampling methods for the metagenomic analysis?
So it was essentially we ran a spin column through just to understand what the contaminants were in the DNA extraction kits that we used, because that's essential with metagenomic analysis, and use that. But we also the metagenomics is actually the unmapped reads, so it's part intrinsic to the whole genome sequencing analysis. It's not because if you think about it, a normal metagenomic analysis is basically just shotgun WGS of a bacterial sample. But in this case, we used the fact that within the sample of DNA we'd extracted, about 10% of the DNA would be derived from bacterial and viral reads, and used that for the metagenomic analysis. Right, it was kind of a byproduct, right? So yeah.
And let me see what else we have here. Someone would like to know about colorectal cancer pharmacogenomics studies. Is that something you've considered at all or interested in?
Yeah, it's very poorly, not poorly done. There's not been much concentration on it. I mean, the classical pharmacogenomics is DYPD, which has been associated with response in 5FU chemotherapy. One of the things that has come out of the 100,000 Genomes project is that, and this project as well, is that because we're sequencing the WGS germline, we get a lot of pharmacogenomic information. So more interestingly, and I don't think I'm betraying a pre-publication confidence, is that 10% of people we found had DYPD mutations, which is pretty important from the pharmacogenomic point of view, because if they have fluorosil-based chemotherapy, they'll have profound toxicity from it. And so, you can get some quite interesting pharmacogenomic insights. We don't understand enough about other tox like cardiac, neurotox, and what the associations are. But one of my collaborators within our institute is studying this in very large colorectal cohorts. And so hopefully we'll have a better idea of how we can identify these things. And using these technologies allows us to highlight these patients. Okay.
Mm-hmm. Can you comment more generally on the use of RNA-seq as a routine method for cancer subtype classification, and what needs to be done to make this more routine?
So I think we need standards whenever you're moving towards whether you call it good clinical practice or ISO accreditation, whatever you have in your own institution, you need some kind of reference material. So the easiest, there are people who've actually made mouse based models for CMS. Owen Sampson has made a mouse cell line based model for CMS, but obviously this is not a mouse we're studying. But for the CRIS classifiers, we've got internally organoid-derived models of each of the different CRIS subtypes we could use for this type of thing. So in other words, you can grow it up as needed as reference material, and they're relatively genomically stable. It's a bit harder with CMS because CMS is based on the entire transcriptional landscape, so the mesenchymal cells, the fibroblasts associated with the cancer. So making reference standards for CMS is quite difficult. I would imagine it would end up being some kind of transgenic model, but we haven't done that yet, but that's what you would need to do, I believe.
Mm-hmm. Okay, thank you. Looks like this is all the question we have for all the time we have for questions today. There are a few more questions that we didn't get to, but this is it. Let me thank our speakers, Dr. Andrew Beggs and Dr. Lucas Paul, and our sponsor Lexogen. And if we didn't get to your question, we will try to have somebody follow up with you via email. As a quick reminder, again, please look out for the survey after you log out to provide us with your feedback. And if you missed part of this webinar or you want to listen to it again, we will send you a link to an archived version via email. So thank you very much again for attending this Genome webinar.